You may know an egg and sperm cell make a baby, but how do we even get egg and sperm cells? Well, the process used is meiosis, not to be confused with mitosis. So what is the definition of meiosis? Well, it's the process by which male and female gametes are made. This happens when one diploid nucleus divides to produce 4 haploid nuclei. Now you might be thinking, what does diploid and haploid even mean? A diploid cell has 2 chromosomes of each type. Think about how humans have 46 chromosomes total. A diploid cell, the starting cell in meiosis, would have 46 chromosomes for humans. As for haploid, they only have 1 chromosome for each type. So in humans, the cells produced by meiosis would have 23 chromosomes. So to recap, meiosis is the process where a cell with a full set of chromosomes divides into 4 cells total, each of them containing 23 chromosomes. There are several steps of meiosis, so here's a basic overview. We'll go over them each individually. It goes interphase, prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2, and finally cytokinesis. Interphase is the stage before meiosis officially begins. This is where cellular organelles double in number, DNA replicates, and protein synthesis occurs. The chromosomes are not visible yet, and the DNA only looks like uncoiled chromatin. Prophase 1 is where the chromosomes become visible and the cell's nuclear membrane disappears. Each chromosome has 2 chromatids, notice the X shape, and prophase 1 is especially important because its events lead to genetic variation. Continuing with prophase 1, first we have homologous chromosomes, and they pair up with each other. So homologous chromosomes are the same size, shape, and genes, but not the same alleles. Think about how people have genes for hair color, but the alleles decide if someone is brunette or blonde. Now, when 2 of these homologous chromosomes are together, this is called a bivalent. The process of them pairing up is called synapsis. Now, why is it necessary for the homologous chromosomes to pair up? This is so a process called crossing over can take place, which leads to genetic variation. A junction is created where one chromatid in each of the homologous chromosomes breaks and rejoins the other chromatid. These random connection points are called chiasmata. The whole process mixes up the alleles, meaning that the final cells will have variation. Metaphase 1 is where the chromosomes become arranged in the center of the cell. Spindle fibers appear on either side of the chromosomes and attach. During this, the pole to which each chromosome is attached depends on which way the pair of chromosomes is facing. This is called random orientation and is yet another factor in increasing genetic variation. Anaphase 1 is when the chromosomes of each bivalent are separated by the attached spindle fibers and pulled towards the opposite poles of the cell. One thing that's important to note is that the chromatids stay together and move together. In telophase 1, once the chromosomes are at the poles, the nuclear membrane reforms around each so that the spindle fibers disappear and the cell becomes 2 cells. And this is an example of cytokinesis. And it is important to note that both cells are still haploid, but each chromosome has 2 sister chromatids. And that process of cytokinesis can be seen here. Now, this entire process sort of begins again with prophase 2. So this time, interphase does not occur and replication of DNA does not occur either. The nuclear envelope breaks down and the spindle fibers appear, and the chromosomes also become visible. In metaphase 2, the chromosomes line up at the center of the cells where spindle fibers attach to each chromatid. In anaphase 2, the spindle fibers pull the chromosomes, this time separating the sister chromatids. These chromatids are pulled to opposite poles. Lastly, in telophase 2, the chromatids reach opposite poles, and the nuclear envelope starts to form and the cells prepare to divide. When the cells actually do divide, this is once again considered cytokinesis. And that's it. In the end, we have 4 non-identical cells to be used for reproduction. These cells are haploid because, as we mentioned earlier, they only have half the chromosomes of the parent. This process continues to happen and is the reason why you're alive today.